Good day, and welcome back. We're going to continue now with our examination of the gastrointestinal tract. Last time we looked at the anatomy from the lips to the pylorus, and now we're going to go back and look at the physiology and the chemistry of what happens between those two portions of the body in the process of digestion. We've already reviewed basically a few of the enzymes that are working. We saw that the amylase, salivary amylase, breaks down carbohydrates in the mouth and in the bolus as it moves down the esophagus, that there's a salivary lipase that breaks down triglycerides, which begins in the mouth but is not completed in the mouth. And then we move down through the esophagus, in which really nothing much happens except transit into the stomach. We want to see now what happens to that bolus in a more detail, almost at a molecular level. So what we're going to look at is a few definitions first. First, there's digestion, which you hear bandied about a lot, and that's really the physical chemical breakdown of gross nutrients into particles that are suitable for absorption, so that we can get those in a small enough particles so that they can be taken into the system. So absorption then is the process of moving those broken down nutrients, usually at a molecular level, across the digestive tract wall and into the bloodstream, where it can be distributed to the cells of the body and taken up by those cells for nutrition. Then that naturally follows into excretion, in which the waste products are brought back into the bloodstream. Those are generally delivered to the kidney for excretion. And then we have products that are not digested at all, which remain in the GI tract. Those go on and move out through the colon for excretion through the rectum. There's another process called detoxification, and this occurs on ingested material that might otherwise be toxic, and we have to render it harmless. So in summary, basically what happens, the mouth begins the digestion, but has no role in absorption at all, and no role in excretion. The esophagus is our conduit, again, no role in absorption or excretion. And the stomach is where digestion begins. Very limited absorption, just water and alcohol. None of these are involved in detoxification. That's really a function of the liver, and we're going to get to that in some time. And then we have two kinds of digestive processes which take place in the stomach. The first is mechanical digestion. What happens is that bolus gets into the stomach. It's usually very irregular, thick, slightly watered down with saliva and whatever else we drink, plus gastric juices. There's a lot of gastric juice. And the waves of muscular peristalsis will grind it and mix it in the various portions, from the cardia to the fundus to the body of the stomach. And at that point, where it's mixed with the stomach juices, we call it chyme. And at this point, it's going to be an acid chyme because it's going to be in an acid environment. Very little of this mixing takes place high up. Most of it takes place down in the body, below the fundus and the body of the stomach. And the salivary enzymes continue to work in the fundus for a while, because remember, they work at a higher pH, and we haven't had enough acid mixing yet, even though the fundus is pouring out the acid from the parietal cells. And then as the bolus moves down, it becomes more and more acidic, and this basically neutralizes the salivary amylase and lipase. So not much more salivary digestion is going to go on once we hit the main part of the stomach. Then we get more grinding and mixing, and so again, a very mechanical process. And finally, in the antrum, there's actually purposeful holdup. Only a small amount of this chyme can get put into the duodenum at a time. It won't, the duodenum is very small, the pyloric opening is very small, and the bolus of food and chyme basically remains in the antrum for additional mixing, additional grinding, and some additional breakdown. And here is where the chemical processes begin. This is where they're really starting to take place. What happens first is that the the parietal cells are the big player here. They secrete hydrochloric acid in very, very high concentrations. And hydrochloric acid has the following functions. And there's a whole list of them. First of all, it denatures proteins. Proteins are chains of amino acids. These are small organic acids linked together by very specific and similar bonds. Every amino acid has one end that has one structure, another end another structure, and they link together, and they can only link together that way. They can't turn around and link this way. It's like a little train. It has to have the right coupling. But in order for proteins to function, and all our processes are carried out by proteins, gene function, all the metabolic processes are modulated, regulated, and carried out by proteins. So proteins have a very, very specific geometric configuration. It's not a long chain. They're intricately folded and twisted and turned and held in those positions by bonds within bonds, and they can only function when they're geometrically correct. I'm mentioning this now because it's going to come up later in lots of other of our discussions. So the proteins are denatured. It means they're made inactive and destroyed in their physiologic properties by the acid by unlinking the bonds and letting them open up. And what that does, it exposes all the little links to other molecular products that are going to break them apart. Now, that's number one. That's the main function of the acid. The acid also kills microorganisms. Our mouth is very dirty. Bacteriologically, the human mouth is one of the worst. You'd rather be bitten by a dog than another person. And they're not all pathogens, but many of them are. They could, in the wrong place, produce disease, as well as the fact that what we carry into our mouth on food from our hands may also be diseased. The acid environment is that first cleansing pool that kills most of the bacteria that go through the stomach. And they don't kill all of them. Obviously, ones that cause intestinal disease can get through. E. coli, salmonella, and even tuberculosis, which has a waxy coat on its outside, protects it from the acid. Lots of bacteria have evolved mechanisms to get around the acid in the stomach. There's one that resides in the stomach wall called Helicobacter, I'll tell you about later, that makes an enzyme that splits urea, that releases ammonia, and it coats itself in ammonia, which neutralizes acid. Very clever evolution to protect itself in that environment. The hydrochloric acid also stimulates the flow of the bile juices, of hormones, of pancreatic juices, and it gets them all ready for release into the small intestine. There's a neurologic signaling going on there that's telling the intestine, I've got a bolus of food, you're going to get it next, get ready. And the intestine starts getting revved up to accept small amounts of the chyme. It inhibits the activity of the hormone gastrin. I'm going to talk about that in a minute, but this is what we call negative feedback. Negative feedback is working all through the intestine. We get a signal sent. This signal goes around and releases one substance, let's say gastrin-releasing hydrochloric acid. Told you about that in the last lecture. The hydrochloric acid goes back and signals the gastrin to stop. When the hydrochloric acid level drops, then the gastrin turns on again, and this is a negative feedback loop. It maintains balance and homeostasis. We look now first at these parietal cells that line the crypts of the body and the fundus and the cardia of the stomach. And then we have the chief cells right here that also are in these crypts. The chief cells secrete pepsinogen. Now, pepsinogen is a precursor. That's why it has the ending GEN, because it gives birth to pepsin. And pepsin is converted, pepsinogen is converted into pepsin by the hydrochloric acid, because we don't want that pepsin living inside the mucosa and digesting it. So it's only active at a very acid pH. The pH inside the mucosa, where it lives, inside that cell, is very neutral, just like the body, almost on the basic side of things. It's when it gets out into the lumen or the cavity of the stomach that things are very acid. And the separation is kept by the mucus, which is produced by the mucus cells. Pepsin breaks down those bonds I talk about between the amino acids that are linking the, in a chain, are making up the protein. So it's been exposed by the hydrochloric acid, the pepsin comes in and snips them off into what are called peptides. And a polypeptide is a short protein. And then these long peptides are the proteins that we're usually talking about. Chief cells also secrete gastric lipase, which is very much like the lipase we saw in the mouth, except the gastric lipase is active at a lower pH or a higher acidity. And it breaks down those triglycerides that we talked about and converts them into a glycerin backbone, which has the 3 stems, and it would each have a fatty acid. That's a triglyceride. It snips off 2 of those, leaves a monoglyceride and 2 chains of fatty acids, which can then be absorbed more easily. We're basically taking the big molecules and making them small molecules so they can get across the capillaries and into the bloodstream, and then eventually out of the capillaries and into the cells. It's really a matter of physical size, except we're moving from gross to microscopic to molecular. You can't see these molecules. Lingual lipase and gastric lipase are basically overshadowed later by the pancreatic enzymes. They produce a pancreatic lipase, and that's going to go on in the small intestine. The mucus cells, as I mentioned, continue to protect the lining of the stomach, and I would say sometimes because there are defects in that mucus which allows us to get erosions and ulceration, which are like sores in the stomach, and penetration and then autodigestion. That's what an ulcer is. The digestive properties start going into the wall of the stomach because of some breakdown in the protective process, and we'll talk about that in a bit. The enteroendocrine cells, or the G cells, which we looked at on the slide and are right here. And as you remember, these are only in the antrum. These cells release gastrin, which can stimulate the release of hydrochloric acid and pepsinogen. Now, we have lots of feedback loops going on. Pepsinogen increases gastric motility. Pepsinogen is going to want to make the stomach contract because it's going to get out there and it knows the digestive process needs activity. It also is going to relax the duodenal sphincter, the pyloric sphincter, and tell it to open up so we can squirt some chyme into the duodenum. Duodenum is the first 12 inches of the small intestine. And at the same time, it closes that lower esophageal sphincter, which prevents reflux back up into the esophagus. So as the stomach squeezes, it is prevented from going back to the esophagus because of the activity of these enzymes and allowed to go further on into the duodenum. That's what happens in the stomach in a gross way. And all this is modulated and controlled by the 3 phases of gastric motility, or the 3 phases of gastric digestion. We really talk about motility. What makes the stomach contract and go on to the digestive process? The 3 phases are very important. They've always been very important to us physiologically, and before the age of these miracle acid blockers, they were very important to the way we perform surgery to try to reestablish a normal physiology for the patient to protect them from gastric and duodenal ulceration. The first of these phases is called the cephalic or the neural phase. It comes from the brain. The cephalic phase is mediated by the vagus nerve. I told you we'd get back to these nerves. Do you remember the nerve number 10, the vagus nerve, is that huge nerve, parasympathetic, runs down to the eyes, the face, the chest, slower down of the heart, goes all the way to all the intestinal organs. It's basically a universal nerve. It's parasympathetic, so this means it's associated with rest and relaxation. And I told you that the parasympathetic system is almost always inhibitory, except in digestion. When you are resting, relaxing, and restoring, you actually have to increase the activity of the gastrointestinal tract. So the vagus nerve is stimulatory in the gastrointestinal tract. The same nerve firing to the heart is going to slow the heart. This same nerve firing to the stomach is going to upregulate it. So what happens when the cephalic phase is stimulated, the vagus nerve will control muscular contraction, so the stomach will grind harder and try to empty. It stimulates directly the secretion of hydrochloric acid from the parietal cells and pepsinogen, and stimulates mucus production at the same time. Because the more acid and pepsinogen you have, you're number one going to wipe off the mucus, and number two, you're going to try to eat up the stomach. So it's going to keep secreting mucus during this active phase. The Pavlovian reflex, as well as our own drooling and salivating when we get to someplace yummy, is all controlled by the vagus nerve. Now, things that will stimulate this are things like the cortical perception of food. If you even think about food in your upper brain, you're going to stimulate the vagus nerve. But remember where it comes out. It comes out in the brainstem. So there are connections maybe from the cortical level, but there are also connections in the hypothalamus and the medulla oblongata in the brainstem. They all go down through the vagus nerve to give this parasympathetic stimulation. Sympathetic stimulation, on the other side of this autonomic nervous system, will do everything the opposite. During your fight and flight, you don't want to be digesting food. You don't want gastric blood flow to the stomach to increase. You want it to go to your arms and your lungs and your heart. So everything I told you about the vagus nerve is the opposite during stress. And that's why it's not a very good thing to have a big meal and then go out and run a marathon. It's going to conflict, and actually what's going to suffer is the sympathetic side, because the blood flow is already going to be going to the stomach. There'll be disastrous effects on the way you feel if you try to do this. So that's the first phase, the cephalic phase of gastric secretion and motility. The second phase is called the gastric phase, and this is both neural and humoral. Humoral means things that are circulating in our blood, and we'll come across that, and we use that expression in other places. This phase is initiated by the very presence of the bolus in the stomach. So the presence of food is not required in the cephalic phase. All you need to do is think about food. Your stomach can be and often is empty, but it's just the sight, the smell, the thought of food is sufficient. However, this phase, the gastric phase, is activated by stretch receptors in the stomach wall. And we talked about very sensitive stretch receptors, that if I took the intestine in an awake patient, stomach or anywhere down, and stretched it, this patient would howl. It's very painful. If I burn it, cut it, put a stitch through it, they wouldn't feel anything. There are no receptors for that kind of pain. But stretch is important to the stomach, and these stretch receptors, when they're not carried out to extremes, are not perceived as pain. They're just noted by the autonomic nervous system, and it's mediated by the vagus nerve because the reflex tells it what to do. That reflex goes up to the brain and says, I've got something in my stomach, I need more gastric juice. And it intensifies the cycle. The food then neutralizes the pH of the stomach. The food's going to come in and sop it up. We all know that when we have that acidy feeling in our stomach, one of the good things to do is to drop some crackers or bread down there, sop it up literally like a sponge, and it neutralizes the pH environment. Well, there are also chemoreceptors in the wall of the stomach that measure pH. And those chemoreceptors detect the rise or the neutrality in pH and signal through the brain that we need more acid because the bolus is there. So think about what happens when, in the old days, we used to take lots of Maalox or Tums. We had a lot of acid in the stomach. It made us feel bad. We pop some Tums or Maalox or other chalky substances that are mostly calcium and aluminum based, so they're very basic. And they go and neutralize the acid. The pH would rise, the stomach chemoreceptors would say, whoa, more acid, and we'd get a vicious cycle. You'd be pumping more and more of this stuff. It would make you feel better for a while. An hour later, you need some more because you've stimulated the signal for the stomach to require to to make more acid and therefore require more neutralization to make you feel better. The enteroendocrine glands, which are secreting gastrin, are stimulated by this stretching, especially in the antrum. When the antrum stretches, it sends out gastrin, says, give me some more acid in the stomach. So you have this second phase. And basically, the total effect of this output is to stimulate contraction and then put a small amount of chyme into the duodenum to get rid of it. So all the receptors together are stimulating the contraction while digestion is going on, and the pylorus will open up a little bit and you'll squirt a little bit of very well-chewed, liquidy chyme out of that bolus and into the duodenum, which will then shut down, because it only wants a little bit at a time. The distension decreases after you get rid of some of this. The pH will drop again with an increase in acidity, and then the negative feedback loop starts all over again. The gastrin shuts the esophageal sphincter, as I said, very, very effectively in patients who don't have a defect with that lower esophageal sphincter. And then we have a third phase of gastric secretion, and it's oddly called the intestinal phase, even though it's regulating what's going on in the stomach. And it's because of things that are happening in the intestine are also feeding back to the stomach. During the entire gastric and cephalic phase, the intestinal phases are inhibitory. Okay, I said that most of the gastric side is excitatory. What happens is the intestines are always trying to slow down gastric digestion and emptying in order to give itself more time to prepare the food, and as it moves along this 28-foot tube, to get absorption, because all your nutrition is occurring in the intestine, the small intestine, which is beyond the pylorus, duodenum, small intestine, until we get to colon. And that on the slide, by the way, on the model, starts about here. Let me give you the big overview. We'll look at this in a lot more detail. I'll take away this liver. Here's the stomach moving down into the pylorus, then the duodenum, which goes that way and has kind of a C loop and ends up coming out here underneath the colon. And then it wiggles around for 28 feet to the bottom of the colon, goes around the body and out the rectum. Where it's behind the colon in that little C loop, and I'll show you in great detail in the anatomy section of that, it's called the duodenum, and it's about 12 inches long. That's what the duodenum refers to. And then the intestine. Not a lot of digestion goes on in the duodenum. Most of that is just transport and more mixing with some more chemicals. Most of it occurs in the small intestine. Now, we get down into the foods that are leaving the stomach and moving on into the intestine. They're going to be mixed up. The intestinal phase of gastric secretion is entirely, during the entire cephalic and gastric phases, as I said, is inhibitory. However, as it moves on, the intestines take a more active role in the actual absorption process, but it's still trying to say to the stomach, give me more time, all the time. Every signal is, the more you put into the intestine's saying, give me more time, I need more time, I need more time. So the gastric emptying, which has been promoted by the gastric distension and the digested protein fragments, the amino acids and so forth, are all being stimulated to move into the intestine. The intestine is sending signals back and saying, slow it down. Lots of things stimulate that emptying that are not part of the normal system, and alcohol and caffeine are two of those, which is why we tend to go into a restaurant and have a drink first, which is why we get up in the morning and have some caffeine. Those all stimulate gastric emptying, which starts to prepare the way for hunger. You get an empty stomach and make it emptier by contracting. That's that grinding you feel, the gurgling, the movement of your intestines in your stomach, which to you feels like hunger. Okay? The hormones which are present and released by the entry of chyme into the intestine also tend to slow things down. So that when you release especially fatty acids and glucose, sugar, into the duodenum, you get something called gastric inhibitory hormone, which is called GIP, because it's a peptide, so it's gastric inhibitory peptide, but we tend to call it gastric inhibitory hormone. It inhibits through the circulation now, not nervous, because it's a hormone. This message goes back and says, slow down gastric secretion, slow down motility. Secretin, another one of the hormones, decreases gastric secretion as well. And there's one called cholecystokinin, which actually refers to the gallbladder. The cholecyst is the gallbladder. Kinin is something that creates movement, kinetic energy. And CCK inhibits gastric emptying. I'll talk more about hormones when we get to the gallbladder. And then there's a neural reflex stimulated by the distension of the chyme in the small intestine, which slows down gastric emptying. Remember, distension in the stomach speeded it up. Distension in the intestine slows it down. When your intestine is full of food, you're not going to feel so much like eating. Things are going to slow down, all those processes, including in the brain. It mediates the sympathetic nervous system and inhibits the parasympathetic nervous system. Okay, it's trying to do the opposite of what the stomach is doing, and therefore this whole phase results in gastric motility and secretory inhibition. Okay, those are the three phases. We mess around with those a little bit in surgery, and I'll explain that in just a minute. The gastric emptying is promoted by the distension, the partially digested protein fragments, and by the other drugs that I mentioned. They all intend to increase the secretion as well as vagal stimulation. And then they lower the pressure waves in the stomach also, just as a mechanical factor. The bolus itself is very important. Duodenal distension, which is in a very small area, the duodenum really is only a bit bigger than my thumb when it's collapsed and relaxed. It can get bigger, but it's a very small organ. Any presence of a bolus in there, presence of fatty acids, glucose, the protein fragments, and other secretions of the enzymes I talked about, secretin and CCK and GIP, these all slow down the stomach. Okay, so you have two basically competing systems, but they are balanced in the normal situation and trying to regulate each other. And we can get that out of whack really easily by overindulging in one type of food or another. Now, gastric emptying is also dependent on the kind of food we put into our stomach. Fats tend to stay there the longest. They're hard to get rid of, they're hard to break down. Proteins are kind of in the middle, and then carbohydrates and sugars remain there the least amount of time. There's something called a satiety index. Satiety is satiation, comes from the word satiation. Satiety is the feeling of fullness or lacking a need to eat. You want to feel satiated at the end of a meal. You'd like not to feel uncomfortable. When people are dieting, one of the things that they want to do is feel full most of the time. They don't want to be hungry. A lot of the diets that concentrate on one food or another at the extreme ends, and I'm really one for moderation in medicine, I don't think you can fool Mother Nature. And when you get to these extreme diets, you're usually doing more harm than good. The best kind of diet and the best kind of eating pattern is something that prolongs the satiety index over a long period of time. Now, we are omnivores, and we are designed and tuned to eat everything, and we are designed and tuned to mix our meals. There was a fad for some time, and may still be going on, where people say it's very bad to mix foods. You should have all your carbohydrates together, and then you should have proteins, and then you should have your fats. And all kinds of things would happen, like flatulence and bloating and unpleasant events, if you mixed your foods together. Well, I don't know how it worked for those people who do that, but the answer is we are still omnivores. We are not designed to separate our food. And it works quite well if you mix them, and even better if you're trying to regulate the amount you have to eat. So if you mix all three components at a single sitting, what you'll get is the carbohydrates rising very quickly in the blood and also telling the brain I'm full very early and then disappearing early. The proteins taking a longer time to get in the blood and tell the brain I'm full and then disappearing mid-range, and the fats taking the longest and staying the longest. That combination of three kinds of foods will give you the most even, steady satiety index. Also, filling your food with your stomach with an enormous amount of food in one sitting makes you feel, first of all, bloated and uncomfortable, but then very rapidly hungry again. Because this overstretched organ, which can, by the way, go from what is here the moderate normal size, this organ can stretch to be hanging way, way down almost to the bottom of your pelvis after something like a Thanksgiving dinner. This organ has enormous ability to stretch. Well, after it's empty again, it collapses up, contracts, and says, I'm hungry. So you may notice that after very big bouts of eating, you tend to be hungry very quickly again. So we have to take a lesson from the grazing animals at this point and say what's really good, and that is to eat. You're much better off eating 6 small, moderate meals per day and graze through the day, keeping the sugar levels in the blood, keeping the amount of food in the stomach and the intestine relatively even, than going way up and way down and way up so that you'll get very hungry again, overeat, get hungry again, and overeat. Diets that for weight loss or for maintenance rather than weight gain that follow those parameters are really by far the best. Let me talk to you a little bit now about, again, what can go wrong. We have a number of diseases that we inflict upon ourselves usually. These are self-inflicted wounds. Alcohol is something that's almost worldwide in consumption. It's certainly artificial, although it happens in nature. There are apples. The marula apple in Africa actually ferments on the trees. Now, I don't know if you've ever seen pictures of animals, elephants particularly, that love this apple. They'll eat pounds and pounds and bushels of these apples, and they'll be standing there and their feet will spread and they'll fall on their chin. And a drunk elephant is really quite something to see. So animals do get drunk in nature on certain types of fermentation. However, man does it the most. And alcohol is readily absorbed from the stomach. It can be very rapidly absorbed from the small intestine. And there is an enzyme called alcohol dehydrogenase. And it breaks down alcohol into water and a resulting particle that is no longer effective as alcohol. It detoxifies the alcohol. We don't have this dehydrogenase very high in our system unless we're active drinkers. Women have much less of the enzyme anyway, so they're more readily affected by alcohol. Generally speaking, women who drink moderately or a little don't have as much enzyme. It'll take much less alcohol at a time than an equivalent amount in a man. And the other thing about this enzyme is the liver does respond to this. So if you're drinking constantly, then you will raise the level of alcohol dehydrogenase, and it will make it easier for you to consume more alcohol. Somebody who doesn't drink is going to get drunk faster by the same amount of alcohol. Other enzymes are also elevated because of the response to toxicity, so good medicines are also inhibited. Therefore, an alcoholic or somebody who just drinks regularly is going to go in the hospital and require pain medication or other medications. They're going to require much higher doses because the person who is not drinking will not break down that medication as readily. I want to talk to you a little bit about vomiting and then about ulcer disease. Vomiting is a response to toxicity, to overindulgence, to excesses in the body. The body just wants the quickest way out, and that is to make you vomit. It's a reversing of the normal peristalsis to bring the food up and out before it gets into the intestine. It's usually secondary to huge gastric distension, or if there's obstruction further down the road, that can also back up and make a person vomit. Blood is one of the most irritating substances in the gastrointestinal tract when it's not in the vessels. So when there's blood in the lumen, central cavity of the stomach, the small intestine, or the colon, it's going to create one of several effects. In the stomach, and sometimes the duodenum, it's going to reverse. That's the shortest way out. So you will vomit the blood. Patient vomiting blood from the stomach will usually have bright red blood or dark red blood coming out. If it's lower down the GI tract, it'll tend to get passed out in the form of diarrhea, and because of the way it's acted upon, it'll come out as a black tarry stool. And black stools usually test positive for blood, and it's just because the blood has been digested. Prolonged vomiting can cause massive derangements in fluid and electrolyte balance because there's a lot of sodium, chloride, potassium, bicarbonate, and chloride ions especially, and hydrogen ions in the stomach. If you lose the acid secreting part, the pH of your body may actually shift to alkalosis. You may have too much bicarbonate. It depends what's in the stomach and what's in the content that you're losing. And then we have purely mechanical problems. There's something called the Mallory-Weiss syndrome. In the Mallory-Weiss syndrome, you vomited so forcibly that right at the gastroesophageal junction, right where the esophagus fits into the stomach, there are very superficial veins, and they're very big veins. We'll look at them in our next lecture. Those veins can actually physically tear. And that patient says what happens to them is that they were vomiting, and then they bent over, and their whole mouth filled with blood. They didn't feel that contraction you usually feel, and the history part of this, you have to take careful history, is that the vomiting came first. They vomited normal food, and then they started vomiting blood, and it felt like it just welled up and poured out of them, not so much the cramps and the gagging of normal vomiting reflex. That's as opposed to someone who has vomiting, blood in their stomach, where the blood is the cause of the vomiting, not the vomiting the cause of the bleeding. And that person will tell you they felt that full contraction, that terrible feeling you get in the back of the throat when you're going to vomit, and they vomited the blood right away. So history taking is very important, detailed, careful examination of what the patient tells you. William Osler, a famous physician, said, listen to the patient, they'll tell you the diagnosis. And it's true. You can make 80 to 90% of all diagnoses just on history. You sometimes have to help bring out the facts, such as that one. Which came first, the vomiting or the blood? Now that's the Mallory-Weiss syndrome. There's another one called Borhave syndrome, which can be much more serious, where the tear is so extensive, it actually tears right through the whole wall of the esophagus, which, as I told you, has no covering on the outside, and empties the esophageal contents into the mediastinum. Mediastinitis is an overwhelming infection that can kill very rapidly. These patients require surgery to repair the tear if possible. They usually staple them together, sometimes to take out part of the esophagus, and to clean out and drain the mediastinum using antibiotics as well. Very serious disease. So vomiting is not without its risks. Let me talk to you now about ulcer disease, because ulcer disease is very interesting and has changed massively. My early career as a surgeon, I probably did 150 to 200 operations per year for complications of ulcers. Those complications include bleeding, obstruction, intractable pain, and perforation. By the time I finished my surgical career, I was doing about one. And that was the advent of real miracle drugs that went to the very basic physiology of ulcer disease. It blocked acid output completely. What we used to do way back then when we wanted to change the physiology is we would try to just work on acid production. Maalox wasn't working, Tums wasn't working, we had nothing else. So we went back in there and we would attack the phases of gastric secretion. First thing we would do is we would go in and we would take out the antrum of the stomach. And we would attach the duodenum to this portion where the stomach was missing. And we would then have gotten rid of, not the acid cells, but as I told you last time, we got rid of the gastrin. There was no more gastrin, and therefore there was no more signaling to the parietal cells. If the disease was very severe, and by the way, this was usually a duodenal ulcer in those days, because the duodenum was very sensitive to acid production. We couldn't take out the parietal cell portion of the stomach very easily, because that would mean sewing the duodenum or the remaining stomach way back up to the esophagus. Very difficult to do. So the next thing we added in patients who were refractory to this is we would take these nerves. Here's the vagus nerve coming through. Remember I told you there was a right and left vagus, but the stomach turned. And now we have an anterior and a posterior vagus. We would come in and cut those vagus nerves and stop the stimulation of the parietal cells. It would also stop the stimulation of gastrin by neural or the cephalic phase. Problem with those patients were this part of the stomach got very floppy. It lost its muscular control and it wouldn't empty. Well, what happens when the stomach won't empty? It gets distended and it signals more acid. So we had to fix that by making the pylorus very, very big. We would cut the pylorus fibers just like we did for that baby, called a pyloroplasty, and then gravity would drain the stomach for the patient. We tell these patients to make sure they remained sitting upright after eating. That was called vagotomy and pyloroplasty, or we could do vagotomy and antrectomy. We did a number of these operations. They all had some problems. We did other operations where we took out most of the stomach and hooked the small intestine back up to the stomach itself. These operations worked actually very well. Then in the last couple of decades, we discovered something else that was causing the ulcers. This is again before we had good drugs. And we found that there was a bacterium that was cultured right out of the center of the ulcer erosion, especially in the stomach, called Helicobacter pylori. And the Helicobacter, as I told you earlier, produces this enzyme, so it protected itself from the acid because it neutralized it. It also protected the bacteria from other host defenses with something called a catalase, another enzyme, and it lived very happily there in the stomach. And all we had to do was treat these patients not with antacids, but many of them with just antibiotics. You could wipe out Helicobacter and the ulcer would go away. Now today we generally do both combined combination. If we make a diagnosis of Helicobacter, you can usually look in the stomach with a scope, take a culture, grow the Helicobacter, and then give them both an acid-reducing medication as well as the antibiotic. The acid-reducing medication that has replaced just neutralization with a base is called a proton pump inhibitor. If you think about what hydrochloric acid is, it's an H+, a little hydrogen atom, and a chloride atom that have separated in solution. Now, without going into physics, a hydrogen atom without its rest of its molecule, like in water and hydrochloride, is a proton. It's only got one proton in the nucleus. That's hydrogen. So these are referred to in the business as protons. That hydrogen ion is made in the parietal cell, and there's an active pump. That pump pumps it out into the lumen of the stomach, and we now have chemicals that are specific proton pump inhibitors. We can take one pill a day and completely turn off all the acid production of the stomach, bring the stomach to almost a neutral pH. We get just enough acid for normal digestion, and yet we make the contents of the stomach so near neutral that when it does reflux into the esophagus, we don't get symptoms of erosion, pain, inflammation, and stricture. These have been an absolute miracle drug. They're very, very non-toxic. They can be used over many years without the patients becoming tolerant to them or destroying them. And they have saved multiple millions of dollars in over time, and probably billions of dollars in the cost of surgery and the complications of surgery. This was really big surgery for these patients that had a high morbidity, meaning bad results, and a significant mortality, and not always a cure. So this has been one of the great steps forward in the use of physiologic knowledge, biochemistry, molecular biology to zero in on one step in the process of disease and knock it right out. Next time we'll continue and we'll move on through the GI tract on our way through the anatomy of this fascinating organ system.